This lecture covers the basics of metabolic regulation. The title slide here is something that you would see if you're in a chemical engineering class. Basically, it's a flow diagram. You have a process, and it usually some sort of a chemical process occurring on an industrial scale. Okay, and you have an input to that process and an output. And usually you control this process by measuring some sort of an output um, on the output stream. And that's fed into a controller, and then the controller can adjust the input into that process based on what's coming out. Okay, so that's in a nutshell. I should also mention you can have a disturbance to the input as well that then goes into the controller, and that can control and manipulate the input as well. Um, but more often you look at a measured output, right? And this is sort of the general scheme that's followed in metabolic regulation. When you have a um, a product that's further down the pathway, it sort of feeds back and controls an earlier step in the pathway. So this lecture is going to cover chapter 15. Okay, the first two sections of chapter 15 are just sort of some general overview of regulation. Okay? And then the third section is where we actually apply these concepts to glycolysis and gluconeogenesis. And I should mention too that we're really not in this class. We're not going to be concerned with the regulation of glycogen metabolism. Glycogen metabolism might be, sort of important to you depending on what you're going to do, but we just don't cover it in this class. I thought really just taking some of that detail and material out would help as far as keeping it a little bit simpler. And that's hopefully what I'll try to do in this class is keep these concepts more basic and simple, and not get so tied up in the details, because this can get very, very complex, as we'll see towards the end of this lecture. Okay, so metabolic pathways, as we've seen, are the biochemical reactions in the living cell. Okay, and they're organized into what we call pathways. These pathways are really sort of man-made in a sense, because what we call a certain pathway are just a limited number of steps that the ones that we're really interested in, right? But there's typically other reactions that occur before our pathway that we're looking at, or after, or come out the sides of the pathway, if that makes sense. Um, Everything in metabolism is connected, as we'll see on a slide here in a second. So when we talk about these linear pathways, we're really only looking at certain reactions that we're interested in. Okay. Okay, the pathways have dedicated purposes. Extraction of energy or storage of fuels are two examples of that. These pathways can be represented as a map, and that's maybe a more accurate way of doing it, showing them all interconnected. Okay. And so this would be the idea of a map, right? And they're color coded to kind of give us a little bit better understanding of, in this case here, I'm assuming this is glycolysis, okay, in the dark kind of bluey purple color. Okay, as we go through that, and this is citric acid cycle, we would consider, this is our pathway, but again, there's other reactions that come in and out of the sides of this reaction. There might be some other reactions, upstream of that we're just not considering when we're looking at, this specific pathway itself by itself. Okay. And as you can also see on this, it is very complicated. Okay, so that's one of the issues in studying metabolism, is how complicated in nature it can get. You block, let's say you have an enzyme, this enzyme, we'll just kind of pick out one. If there was a problem genetically in that enzyme, right, what would you expect? Well, you'd expect that substrate for that reaction, this represented by this dot, you'd have a buildup of that, and you might have, a smaller amount of this product from that reaction if that enzyme wasn't working. Well, that's very easy to see that in sort of a simplistic view. But also what happens is, okay, if you have more of this, that's just going to push into these other pathways that are, connected to it. So you might have an increase here, but you'll also could see an increase of this metabolite or this metabolite. Okay, so it does get a little bit complex. Right, the cell tries to maintain a steady state of metabolites in itself, and that's known as homeostasis. Okay? And what homeostasis is the concentrations of at least most metabolites are what is known as steady state. They don't change. So maybe the concentration, if you're looking at a certain metabolite, you have reactions that are using that metabolite, so it's getting used, but then there's also reactions that are making it. And so the rate of it getting consumed and the rate of it being made are equal. Okay. Pathways tend to be at steady state unless they're otherwise perturbed. Okay. After they're perturbed, a new steady state will be established. The flow of metabolites through a pathway is regulated to maintain homeostasis. Okay? And it's not regulated necessarily at every step, okay, in a pathway. Usually there's maybe one or two steps in a pathway that are regulated to maintain a steady state of concentrations of the intermediates. Okay. Sometimes levels of a required metabolite have to be altered very quickly. And some examples of when that might be the case, you might need to increase the capacity of glycolysis when you're doing some sort of action, like you're running, let's say. So then the capacity of your glycolytic system has to be increased. Okay, and conversely, if you stop doing that action, then the capacity of glycolysis needs to be lowered. Okay? And then after an action as well, your gluconeogenesis, the capacity of that needs to be increased. Okay? And the way to kind of switch back and forth of those, sometimes you have to do that pretty rapidly. If you're getting chased by a lion and you have to run away really quick, you have to have your glycolytic capacity has to turn on really quick and increase. And then when you're stop doing that, you have to recover from that, and glycolysis then has to be turned off. Gluconeogenesis has to be turned on. Okay. One way in this how cells regulate these pathways is by something known as feedback inhibition that we sort of alluded to on the title slide. Okay. A similar block diagram is shown here, where you have a certain process that has inputs and outputs. So in biochemistry, we'd think of these as substrates, and this is products, and this is your enzyme, the reaction your enzyme's catalyzing. A lot of times those products are going to feed back and actually inhibit the enzyme. Okay. Example of that is the fact that ATP inhibits the commitment step of glycolysis. So that's step 3 in glycolysis. Okay. Another example of feedback inhibition is in amino acid metabolism, which we really don't cover in this class, but this is a good example of feedback inhibition where you have a pathway where threonine is converted to isoleucine, and isoleucine then actually feeds back and inhibits that first enzyme in the pathway. Okay, so feedback inhibition is something that you see examples of over and over again in metabolism. And so that's a pretty important concept in regulation. Okay. So how do you control the flux and the overall flow through a pathway? And you do that by altering either the rates of the biochemical reactions, or you can do that just by the presence of enzymes. Ultimately, you're changing, you're regulating and adjusting rates of reactions to control fluxes through pathways. Okay. And so the rates of biochemical reactions can depend on many factors. Okay. The concentration of reactants and products can be a factor in determining rate. Okay, so if you think back to the rate equation, general rate equation, if we have the reaction A plus B going to C, right, that rate's going to depend on the rate constant, but also on the concentrations of the reactants. Okay. So how available the reactants are can influence the rate. Okay. That doesn't always offer much in terms of control, because the cell might not have the ability to control the concentrations directly of the, in this case, we'll call them the substrates to the reaction. So that might not be a primary way of cellular regulation of controlling the rates. Okay, some other ways of controlling rates. One is the activity of the catalyst, right? And this is biochemistry. So what do we mean by catalyst? Well, that's the enzyme. Okay, so you can control the concentration of the enzyme that's present. Okay, and that kind of goes to the rate that the enzyme is translated versus the rate that the enzyme is degraded. Okay, so how much enzyme is around. The other way you can control that is by just the intrinsic activity of the enzyme. Okay, so that enzyme can be affected a number of different ways. Okay, it could depend on substrate, certain effectors, or even phosphorylation sites. And if you think back to the equation for the rate constant now, right, the rate constant depends on a constant times e to the power of minus EA, which is the activation energy, divided by RT. Right? So if we're changing the intrinsic activity of a enzyme, we're altering that activation energy that you get from the enzyme. So that would be how it affects that rate constant. Okay. Right. You can have regulation in terms, it kind of extends from this concept. You can regulate that activity based on the concentration of effectors that are around. So what kind of effectors are there? Well, you can have allosteric regulators, you can have competing substrates, or you can actually have a change in something like the pH or the ionic environment that an enzyme's in. Okay. Let's focus on the first two of these. So if you have an allosteric regulator, that regulator is going to bind to another part of the enzyme that the substrate doesn't bind to, and either make that enzyme more active for the substrate or less active. And in this case, in this example, you have an inhibitor there that's binding, and it's sort of turning that enzyme off, so it's slowing it down. You can also have a competing substrate, right, that binds in the active site and blocks that the substrate binding. Okay, so those are ways that other molecules can kind of adjust the activity of an enzyme, and thereby affecting the rate of that reaction. Okay, and then finally, temperature. Probably not necessarily a major source of control, at least in metabolism, right? Because organisms tend to be fairly constant in their temperature, but if you adjust the temperature, right, just in that same equation for the rate constant, you can see that temperature factors into that equation. So if you adjust the temperature, you're going to change the rate constant and therefore the rate. Okay. So if we look at the life of an enzyme, okay, there's 10 different places in this figure that you can control how much of an enzyme is produced. And all of these places are potential places for regulation or control. Okay. So first, you need to have some sort of an extracellular signal, typically, that comes in either directly into the nucleus or hits a receptor in the membrane, and then that triggers a transcription factor to go into the nucleus. Okay, so the extracellular signal telling the cell to make that enzyme. Then second, you get the transcription of a specific gene for the enzyme. So the DNA is being converted into RNA, messenger RNA. Right? The messenger RNA then has to be translated into protein. Okay? And one way you can kind of control how much of a protein is made is by either the stability or the lack thereof of the messenger RNA. Okay, you can be transcribing a gene, but if that messenger RNA doesn't stick around long enough, you're not going to be expressing much of that protein. Okay, so the messenger RNA can be degraded before much of the protein is made. So that's one way to regulate it. Okay, the other way is actually the translation. Okay, so how much of that messenger is translated, that can be a method of regulation. Okay. After that step, the enzyme is produced, and you have an enzyme that's in the a fully folded protein. Okay, and from there you can have 6 other ways of regulating that. The fifth point of regulation here is that protein can be degraded. Okay, so you can have an enzyme, and then the cell can actively degrade it to get rid of it. You can have that enzyme be sequestered into some sort of a subcellular organelle. Let's say you need that protein or that enzyme to be in the endoplasmic reticulum to be active. So the act of putting it into that endoplasmic reticulum is a way to regulate the rate of the reaction that it can catalyze. Okay. The product binding with that enzyme, that is another point of regulation. A allosteric effector binding it can be a point of regulation. The phosphorylation site or dephosphorylation to either activate it or inactivate it, okay, that can be a point of regulation. Kinase proteins will phosphorylate, and phosphatase proteins generally remove phosphate groups. And then finally, the enzyme that you have can combine with another regulatory protein. Okay, so that can be yet the final little point of regulation. So you can see that there's many different points of regulation to a given enzyme. Now, not all enzymes undergo all of these points of regulation, but you can see that you can have many different levels of regulation. So this can get very complex. Okay. So here we have a figure from an insulin pathway, right? Insulin binding will be turn on a number of different steps, but one is to put a glucose transporter, make that active in the membrane to get more glucose into the cell. The other two things that you see in this figure, it's turning on hexokinase, right, which is the first enzyme in glycolysis. And it's also turning on an enzyme called glycogen synthase. Okay, so this is if we're looking at which points of control do you see in this pathway, well, there are three possibilities here. The fact that it turns on hexokinase and glycogen synthase, that is on the transcriptional level. It actually makes transcription factors that increase the concentrations of those enzymes. And then with the glucose transporter, those are generally already transcribed, but they're sequestered into vesicles. Okay, when you get that signal from insulin, then it releases those back into the membrane where they can transport glucose. So if we look back here, right, we have examples of the transcriptions of genes for hexokinase and glycogen synthase. Right, so that would be, what was that, 0.2 for both of these steps, and then actually getting the glucose transporter into the membrane, that would be an example of here 0.6, where you have the transporter would be sequestered away, and this signal from insulin puts it in the membrane where it needs to go. So if we're going to look through some of these points of regulation in a little bit more detail. Okay. Proteins have a definite or finite lifespan. They don't last forever. Okay. In different proteins in the same tissue have very different half-lives. Okay, some can be less than an hour to even about a week for liver enzymes. If we look at typical liver enzymes, the stability seems to correlate with the sequence at the end terminus of a protein. Okay, some proteins are basically as old as you are, and the examples of those are much fewer examples of those, but one is the crystallins in the lens of your eye. That's an example of a protein it's basically as old as you are. Okay. But by and large, most proteins have a much more finite lifespan, and they're much shorter lived. And just an example here, we have different tissues and the average half-life of the proteins in those tissues are given here. And so most are, a day or so, maybe up to a week. It's kind of, the typical range of an average protein. Okay, phosphorylation of enzymes affects their activity, and we've seen examples of this in Chapter 12 in signaling pathways. Okay. Phosphorylation, again, is catalyzed by protein kinases, and dephosphorylation is catalyzed by protein phosphatases. Okay. It can also be spontaneous, but spontaneous reactions in biochemistry, there aren't a whole lot of examples of those, so we'll focus on dephosphorylations being caused by phosphatases. Okay. Typically, proteins are going to be phosphorylated on hydroxyl groups, and remember, amino acid side chains that have hydroxyl groups are serine, threonine, and tyrosine. Okay. So here we have a protein that can be phosphorylated, and it has a free hydroxyl group. Okay, the kinase will take ATP and take the gamma phosphate from ATP and stick it onto that enzyme or protein. And then the phosphatase protein will do the opposite reaction, but instead of taking that phosphate group and using it for something, it just hydrolyzes the phosphate off. Okay, enzymes are regulated by other regulatory proteins. Okay, and this example here is an example of how you can have a catalytic subunit and different regulatory subunits that bind to it that will give this catalytic site a different activity for, in this case, it'd be different substrates. Okay. So maybe you have the catalytic subunit that's always expressed, but then under different conditions, you would be expressing different regulatory subunits. The rate of the reaction is going to depend on the concentrations of substrates. So we saw this as point number 7 in that figure. Okay. The rate is more sensitive to concentration when you have lower substrate concentrations. Okay. And in that case, the frequency of substrate meeting the enzyme really matters. Okay. So what that means, it's more sensitive, right? At lower concentrations here, we're looking at ATP concentration, and this is the velocity of the reaction, right? At lower concentrations, the rate goes up and down quite a bit. Okay, when the concentrations are higher, the rate doesn't change as much. Okay, so here, if from going from like 1 to 5, you can see that the rate jumps up a bunch, a really big jump. But if we're going from, even 30 to 40, so jumping up 10 millimolar in concentration, that rate doesn't change a whole lot. Okay. So it's more sensitive when the substrate concentrations are lower, less sensitive when the substrate concentrations are high. Okay, and that's because the rate's becoming, the enzyme, the active site on the enzyme is becoming saturated with substrate at higher concentrations. Just a little kind of walk back to some of the enzyme kinetics, right? If we look at the Km versus metabolite concentrations, many enzymes have Kms that are near or greater to the physiological concentration of their substrate. An example that enzymes that don't follow that, those that utilize ATP or NAD are sort of exceptions to that rule. And I guess one way you can maybe rationalize that is that if your physiological concentration of your substrate is near your Km, you're going to be in a fairly dynamic range where the rate of your reaction will change depending on changes in that substrate's concentration. But for example, for ATP or NADH, if you look at the plot here, and those are going to be in these kind of orange squares and red squares and blue squares, they tend to be in a region here where the metabolite concentration is higher than the Km values. And so there would be maybe a little bit further up on this chart. So maybe the concentration, if your ATP or NAD, NADH concentrations change a little bit, it's not going to affect that overall rate of the reaction as much. Okay. Reactions that are far from equilibrium are common points of regulation. Okay? When a metabolic pathway operates, they operate in very close to equilibrium. Okay, and the key enzymes in those pathways are operating far from equilibrium. Okay, so most reactions in a pathway are in equilibrium, but then the key steps in the pathway are far from equilibrium. Okay, those are the sites for regulation, and those are the places where you can control flow through the pathway. Okay. To maintain a steady state, all enzymes in a pathway have to operate at the same rate, right? If you had enzymes that are catalyzing reactions at different rates in a pathway, right? If one step's going much faster than another step, you can kind of see how that would be sort of a bad thing. You're going to deplete one part of that pathway and have a buildup on another part. And so then the example that's shown here, the just generic example, we have a pathway where A goes to D in 3 steps. Okay, and then we can see that the first step, we're given rates, velocities for the forward reaction and the reverse reaction. So for all of these, we're given a forward velocity and a reverse velocity. Okay. What you'll notice here is, okay, which step do you think is regulated? Well, if we look at the rate of the forward and reverse reactions of all of these, for the second and third step, those forward rate and reverse rates are very close to one another. Okay. The rate of the forward reaction and reverse reaction on the first step are orders of magnitude different. Okay, so the first step here would be, the candidate for regulation of this pathway. Okay, the net rate of all of them is the same. It's 10. So if you just take the rate of the forward reaction minus the rate of the reverse reaction for all of these, it all equals 10. So the rate through the pathway, the flux through the pathway is consistent. Okay. We're not going to ever, if this was 20 and these were 10, you would get a buildup of B here. Okay, so these are all equal, so the flux through it is controlled. Okay. Right, table 15.3, you don't have to memorize any of these numbers, but this kind of shows you an example of if we pick out some reactions in glycolysis, maybe we can find the ones that are closer to closer to equilibrium. Those would be the reactions that aren't regulated, and the ones that are not close to equilibrium, those are going to be the reactions that are regulated. Okay, so if we look at these, hexokinase, that's step 1, PFK1, that's step 3, those are not near equilibrium. So those are going to be points of regulation. And then also pyruvate kinase, the last reaction of glycolysis, okay, not near equilibrium. And then finally, the pyruvate carboxylase and pep carboxykinase, that would be the first step of gluconeogenesis, again, not near equilibrium. Okay. And we sort of mentioned that in the lecture on glycolysis and gluconeogenesis, right? We mentioned those reactions, and we sort of mentioned those are the regulated steps, and that's what we're seeing here again. Okay. ATP and AMP turn out to be very key regulators in metabolism. 10% decrease in ATP can greatly affect the activity of ATP and enzymes that use ATP. Right? 10% decrease in ATP leads to a dramatic increase in AMP. And that's why sometimes you'll see AMP is actually the metabolite that is an allosteric regulator. And here's an example of some concentrations of these nucleotides before and after a ATP depletion event in a cell. So the concentration in this typical cell would be 5 millimolar ATP, 1 millimolar ADP, and then 0.1 millimolar AMP. Okay, and then after some event that ATP is consumed, the concentration of ATP might drop 10%. Okay, so go from 5 millimolar to 4 millimolar. ADP concentration here is staying the same. AMP concentration goes from 0.1 millimolar to 0.6, which doesn't seem very like the concentration's very high, right? But that's a 600% change. Okay, so ATP concentration, because it is typically higher, right, you can have a 10% change in that concentration, and that leads to a 600% change in the AMP concentration, right? And so that's why AMP is really can be more potent of a regulator, because its concentration can vary by a much wider margin, because it is typically in lower concentration. Right. AMP differently affects pathways in different tissues via something known as AMP-dependent kinase. Okay, so if you have increase in AMP concentrations, this in the brain might signal something that wants you to eat, makes you hungry to intake food. In the heart, it turns on fatty acid oxidation, glucose uptake, and glycolysis. In fat tissues, it turns off fatty acid synthesis. In liver tissues, it'll turn off fatty acid synthesis and cholesterol synthesis, because if you have higher AMP and lower ATP, you're at a state where your body needs energy, so it doesn't make sense to waste energy to build up fatty acids or cholesterol, right? You need to be breaking them down for energy. Okay. In pancreas, it'll turn off insulin secretion, and then skeletal muscle, it increases the fatty acid uptake, oxidation of glucose, oxidation or mitochondrial biogenesis. So it's increasing your ability to burn up, to burn things and make energy. Okay. So don't need to know any of these specifically, but this is just an example of how one signal, and a very potent one, AMP, can have different effects in different tissues. And that's sort of an example of, we talked about it in chapter 12, how the same signaling molecule can have different effects depending on the tissue. And this is an example of that. Okay, some enzymes in the pathway limit the flux of metabolites more than others. Okay, so enzymes that are far from equilibrium, those are the ones that are regulated. Okay, not all regulated enzymes have the same effect on the entire pathway. Some will control the flux through the pathway, which is how much product is ultimately made at the end of the pathway. Others regulate steady state concentrations of metabolite in response to changes in flux. Right. Hexokinase and phosphofructokinase are appropriate targets for regulation of flux through glycolysis. Okay. Increased hexokinase activity enables the activation of glucose. Increased PFK1 activity enables the breakdown of glucose via glycolysis. Okay, so those are the two enzymes that really control the flux through that pathway. Okay. And particularly hexokinase affects the flow through glycolysis more than PFK. Okay, so here we have a figure that shows the the x-axis is the amount of the enzyme is added and how much glycolytic flux you get. So adding more hexokinase really has a big effect on the amount of flux through the pathway. Adding more PFK1 will have an effect, but not as great as hexokinase. And then an example of an enzyme that isn't regulated, okay, phosphohexosisomerase, you add more of that and nothing. If you're just adding more of that enzyme, that doesn't increase the flux through glycolysis at all. Okay. So the second section in chapter 15 talks about a few terms. One of those is called the flux control coefficient. Okay, this shows the relative effect of each enzyme has in setting the rate through the pathway. Okay, so these coefficients, they're really dependent on what you're defining as your pathway, and however many reactions you include in your pathway, the flux control coefficients all have to sum up to 1. Okay, so they're sort of an arbitrary value in that sense. Okay. So if you look at all of these, in this our pathway is going to be defined as A going to D here. Okay, so the flux control coefficients we see for the pathway are given here, right? So the step that has the most control over that flux is going to be this step, right? Because it has a higher flux control coefficient. Okay, this one is 0, so this one is not a regulated step. It's in equilibrium. Okay, this one has a flux control coefficient, but it's smaller, right? So this is a regulated step. It does control some flux, but not as much as this step. And then if you have a side pathway, a place that breaks off from the what we define as our main pathway here, this becomes that's why that it's a negative value there. Because if you increase this reaction, right, it's going to pull off more and eliminate some of that product that you're forming at the end of your pathway. Okay, so that's the basics of these flux control coefficients. Okay, the most important thing to remember about them is when you're looking at these, they have to sum up to 1. Okay, there's also something called the elasticity coefficient. Okay, and that measures the responsiveness to a substrate. Okay, so if we're plotting the substrate concentration versus our rate of reaction, and we do that typical Michaelis-Menten plot, when our substrate is low and we're getting a pretty high response to that substrate, that elasticity coefficient is basically 1. Okay. When you conversely have a high substrate concentration, okay, that rate is not very dependent on that substrate concentration. The elasticity coefficient there is 0. So these will vary. Elasticity coefficients sort of vary between 1 and 0. Okay, and then finally we have the response coefficient, which is capital R. Okay, and this is defined as the responsiveness of a pathway to an outside factor that affects certain enzyme in the pathway. And really that is, it's defined as the flux coefficient times the elasticity coefficient will give you the response. Okay, and that's really chapter 15, section 2 in a nutshell. On an exam, you would have to be given, these elasticity coefficients, and then the only way you'd either have to be given that flux coefficient, or you'd have to be given some sort of chart like this with all of the values except 1, basically, and then asked to calculate the the missing flux coefficient. And then if you're trying to calculate a response coefficient, right, it's just flux coefficient multiplied by elasticity coefficient. All right. So now we can talk about, that's the basics of metabolic regulation. Now we can kind of focus in on some specific ways that glycolysis and gluconeogenesis are regulated. Okay, so just remind ourselves, we have glycolysis and gluconeogenesis. Glycolysis, those important steps that are regulated again are step 1, hexokinase, step 3, phosphofructokinase, and then step 10, pyruvate kinase. And then with gluconeogenesis, the regulated steps that we'll talk mostly about, the first 2 steps here regulated. One that we'll mention quite a bit about is the fructose 1,6-bisphosphatase. That step, which would be the partner to the third step in glycolysis, that we'll also talk about the regulation of that. Okay. And this figure I took from the internet is a little bit better of a summary on how these certain steps are regulated and some of the things that regulate them. Okay, so if you, I won't go too much into detail with this, but you'll see as we talk about this in some more detail, this step 3, right, how it's differentially regulated. A molecule called fructose 2,6-bisphosphate, or AMP, will turn that enzyme on towards glycolysis, that is. And then other molecules will turn on gluconeogenesis, such as citrate. So this kind of just gives a good overview, if you will, a good way of summarizing how these 2 pathways are differentially regulated. And we've seen in this lecture already how hexokinase does affect the flux through glycolysis. Okay, so that's the first step in glycolysis. Hexokinase really has a big effect on the flux through that whole pathway, even more so than PFK1 does. Okay. So there are 4 what are known as isozymes of hexokinase. Okay, so what an isozyme is it's a sequence of a protein that's a very similar sequence, but usually it's slightly different. It has its own gene. Okay, so they're very closely related sequences, but they are different protein sequences. Okay, and they're encoded by different genes. Okay, they're catalyze the same reaction. Okay, their catalytic or kinetic properties may be different, and they can be regulated differently. And a lot of times they're expressed in different levels in different tissues. So one isozyme of an enzyme might be expressed in liver, and another isozyme of an enzyme might be expressed more in the heart or in the brain. Hexokinase 1, the isozyme hexokinase 1, is expressed in all tissues. There are different levels of expression, but it's expressed everywhere. Hexokinase 4 is only expressed in the liver. Okay. And hexokinase 4 has different kinetic properties. One of the differences is it has a higher Km. Okay. So what does that mean? It means it's responsive to a higher glucose concentration. Okay. It's also not inhibited by glucose 6-phosphate, so it can function at higher glucose concentrations. And it functions to clear blood glucose at higher glucose concentrations for storage as glycogen. So you can think of that as being specific to the liver, right? When you get really high blood glucose, um, normally hexokinase wouldn't be active, right? Because you don't want to, if you have a bunch of excess glucose and you don't need the energy, you don't really want it to be broken down through glycolysis, because what ultimately will happen there is you'll continue on to the citric acid cycle, and you'll get too much acetyl-CoA, and that'll build up and go to fat. Um, so you kind of want to stop, right? So you wouldn't want to be wasting uh, glucose when you don't need the energy. So typically hexokinase you would think of not wanting to operate at those high glucose concentrations, but in the liver, um, you want the liver to be able to act on higher blood glucose concentrations, to be able to get that glucose out of the blood into the liver cells where it can be converted into glycogen. Okay. And so that's why hexokinase 4 has these kinetic properties that allow it to work at a higher glucose concentration. Right. So just shown here is kind of what we're talking about with hexokinase 1, right? As you can see that it has a very low Km, so it's not going to be it's going to basically be at a steady state at the higher glucose concentrations. Hexokinase 4, however, has a larger dynamic range, right? It has a higher Km value, so a higher dynamic range that it can, adjust its rate of catalysis. And I should mention that these are relative enzyme activities here. So it looks like hexokinase 1 here is much higher in activity, but those are, again, relative enzyme activities. Right. So hexokinase 4, even at 10 millimolar glucose isn't close to its highest activity yet. And so this is probably a better picture of showing this. Hexokinase would be hexokinase 1, and this glucokinase is actually hexokinase 4. So that's maybe a little bit confusing here, but right, glucokinase is just that fourth isozyme of hexokinase. And so these are shown a little bit better to scale. Okay, so at lower concentrations of glucose, hexokinase 1 is active, and the activity of hexokinase 4 is lower. But as you get higher and higher concentrations of glucose, then you can see that the activity of glucokinase continues to increase. Okay, whereas hexokinase 1 it reaches a maximum. Right, and so this figure also shows you the concentration of fasting blood glucose. Right, in fasting times, the hexokinase 1 that activity is winning out, but as soon as you eat, right, and get some blood sugar, that's when the hexokinase 4 activity is going to start dominating. Okay. Hexokinase 4 is regulated by sequestration and transcription. So if we think back to those points of regulation, those are, I believe, if I remember correctly, point 2 is transcription, and point 6 was sequestration. Okay, so how does this happen? Well, when glucose comes inside the cell, right, that creates activates sort of pathway that helps hexokinase be expressed in the nucleus. Okay, glucose also being converted to fructose 6-phosphate in glycolysis, that actually feeds back, and once that happens, you get the opposite effect. That hexokinase is sort of sequestered back into the nucleus, okay, by the addition of a regulatory protein. Okay, so in that case, you have both transcriptional regulation and the regulation of sequestration into the nucleus. Okay, step 3 of glycolysis, and this is probably the one we'll focus most on in terms of regulation, and this is the step that's catalyzed by phosphofructokinase 1, or PFK1. Okay. That reaction, again, is fructose 6-phosphate going to fructose 1,6-bisphosphate. Okay, and that was the commitment step to glycolysis. After that point, fructose 1,6-bisphosphate is committed to going all the way through glycolysis. Okay. This is a very interesting enzyme, PFK1, because it actually has 2 different sites for ATP. Okay, ATP is a substrate of this enzyme, but it's also a negative effector. Okay, so it has one site that binds ATP as a substrate and another site that binds it as an allosteric effector. Okay, and really why you want to regulate this step is you don't want to spend glucose in glycolysis if there's already plenty of ATP around. Okay, you want to save that glucose in the form of, primarily in the form of glycogen, to be brought back out when you need it. Okay. So our reaction is fructose 6-phosphate plus ATP going to fructose 1,6-bisphosphate plus ADP. Okay, remembering back. Okay, the things that affect this reaction, okay, ATP negatively affects it, okay, turns it off in a sense. And sort of the opposite of ATP would be AMP or ADP, right? When those nucleotides aren't fully phosphorylated, that turns this reaction on, right? Having a higher concentration of AMP in the cell is telling you that the cell needs energy. So you turn on glycolysis. Okay, having more ATP around, okay, that tells you, okay, our energy requirements are met. We can turn this off. Okay, the other things that maybe aren't so obvious, right? Because these are, you can think of this as a feedback inhibitor, because glycolysis is used to produce ATP, right? So it feeds back and turns it off. Okay, some of the other things that might not be so obvious, one is citrate or citric acid. That will turn this off. Okay, and that makes more sense when we talk about go through the citric acid cycle. Okay, citric acid cycle, right? Citrate. Okay, if that's going and has enough substrate for the citric acid cycle, you don't need any more things being pumped into it, so you turn off glycolysis. Right. And the other one that might not make any sense is this molecule called fructose 2,6-bisphosphate. Okay, that turns this on. Okay, and a very important distinction here is that fructose 2,6-bisphosphate is not the same as fructose 1,6-bisphosphate. Okay, fructose 1,6-bisphosphate is an intermediate in glycolysis, so it's going to go on to the next step, the next enzyme. Okay, it's a substrate for the next enzyme. Fructose 2,6-bisphosphate is not. It's not an intermediate. It's not a product. Okay, it's a regulatory molecule. Okay, an allosteric regulator of PFK1. And we'll explain this a little bit more. Okay, so this is a picture of PFK1, and you have certain sites here where you have an allosteric regulator of ATP, and those are shown here. Okay, so this is kind of a tetramer. There are 4 subunits in PFK1. Okay, there's an allosteric ATP site, and there's also a catalytic ATP site, so that would be in here. Okay, they have ADP being bound in the crystal structure, but that's where ATP would come in. Okay. And then this is the other part of the active site where you produce your fructose 1,6-bisphosphate, shown here in yellow. Okay, so you have allosteric sites for ATP and a catalytic site for ATP. Okay, and that's what makes this enzyme pretty unique. The affinity for ATP, if you're thinking, well, which site has more affinity for ATP, and that would be the active site. So when ATP concentrations are low, it can still bind to the active site. You want it in that active site. As that concentration of ATP gets higher, then it can start binding into the allosteric site that inhibits the enzyme. Okay, and that's kind of shown in this, sort of shown in this figure, right? You have the activity of PFK1 at high ATP in red and low ATP in black, right? And this is the substrate concentration. So at high substrate concentrations, you have, at high ATP concentrations, right? The adding more of your substrate makes the rate go up slower and slower, okay? Or the rate doesn't go up as fast as when you have low ATP, right? So that means that ATP is acting as an allosteric regulator, right? So when you have low ATP concentrations, your enzymes really binds that substrate really well. Okay, when you have high ATP concentrations, it doesn't bind your substrate as much. Okay, so that really makes this unique in that it's a substrate that also is acting as an allosteric regulator. And as we mentioned before, the amount of ATP and AMP are really a good indicator on the energy requirements of a cell. Okay. And so this figure sort of shows that. I got this out of a paper. But you'll have ATP levels either being consumed and turned into ADP. So for ATP consumption, examples of that would be, things like cell growth or the activation of motor proteins in like a bacteria, for example, for it to move, right? It needs to have that motor protein that will consume ATP. Okay, so ATP is consumed, goes to ADP. In catabolism, you're forcing ADP and converting it back into ATP. Okay, some things that will block catabolism would be metabolic stress. Okay, more important for our class though is the idea that ATP gets converted to ADP. So then, why is AMP the one that the concentration changes? That's because there's an enzyme called adenylate kinase, and it takes two ADPs and it makes one AMP and one ATP. So that way it sort of recycles it to get the most amount of ATP out as it can. Okay, the most bang for your buck for ATP. Okay? And so that's why the ADP concentration is sort of constant and AMP concentration can change as ATP is used. Okay. So if we look at this regulation of PFK1, and also conversely fructose 1,6-bisphosphatase in gluconeogenesis. Okay, these enzymes have to be sort of regulated together, right? Because you don't want to turn them both on at the same time and create a futile cycle. Okay. So in general, you want to go glycolysis if AMP is high and ATP is low. You want to go gluconeogenesis if AMP is low. Okay. So that, in a nutshell, this is how these are regulated. So PFK is the enzyme for glycolysis, right? ATP inhibits it, ADP and AMP turn it on, and then also citrate inhibits it. Okay. And then you can think of AMP as turning off the gluconeogenic enzyme. Okay. Right. So in liver, fructose 2,6 bisphosphate it becomes important. Okay, this again is not a intermediate of glycolysis. It's only a regulator. Okay. It's produced specifically to regulate glycolysis and gluconeogenesis. Right. What fructose 2,6 bisphosphate, or F26BP, as it's sort of abbreviated sometimes, what it does is it activates PFK1 in glycolysis and inhibits fructose 1,6 bisphosphatase in gluconeogenesis. So this one molecule can activate glycolysis and inactivate gluconeogenesis. Okay. So if we look at the rate of, in this case it's PFK1, so glycolysis, in the presence, by varying the concentration of fructose 6-phosphate, its substrate. Okay, so without any fructose 2,6 bisphosphate, without any of that around, you can see that this blue curve shows you that this enzyme it doesn't bind its substrate as tightly, and the rate is much lower. Okay. When fructose 2,6 bisphosphate is present, the binding of the substrate by PFK1 increases greatly, which increases the rate at those lower substrate concentrations. Okay, so F26BP is turning that PFK1 on. Okay, it's turning glycolysis on. Conversely, when we look at the gluconeogenic enzyme for the same that same step, in the presence of F26BP, again that is making this rate binding of the substrate and the rate of it much lower than when it's not present. So it's an opposite effect here. It's turning off gluconeogenesis and it's turning on glycolysis. Okay. So if we look at the effect of F26BP, it turns on PFK1, it turns off FBPase1. So turning on glycolysis, turning off gluconeogenesis. Okay, and this is probably one of the most important regulatory molecules, because it makes, as you can kind of see in this figure, it makes a huge difference on the rate of a reaction. Yeah. Okay. Now where it gets really confusing, how is F26BP, fructose 2,6 bisphosphate, how is it produced? Well, it's produced from fructose 6-phosphate. Okay, fructose 6-phosphate is a intermediate of glycolysis. Okay, some of that doesn't go through glycolysis is used to produce fructose 2,6 bisphosphate. Okay. It's catalyzed, this cycle is catalyzed by enzymes that have very familiar names, and this is where it can get very confusing. Okay, converting fructose 6-phosphate to fructose 2,6 bisphosphate is done by an enzyme called PFK2. So phosphofructokinase 2, okay? Not to be confused with PFK1, that's the glycolytic enzyme. It's a different enzyme called PFK2. Okay? And again, that uses ATP in that process to add that second phosphate group. Okay. It's dephosphorylated by fructose bisphosphatase 2, FBPase2. Okay. Again, not to be confused with the enzyme in gluconeogenesis, FBPase1. Okay, that's a different enzyme. And it turns out these two enzymes are actually part of the same complex. One side of it has a PFK2 activity, and the other side has the phosphatase activity. Okay. So how do these work? Well, it depends on phosphorylation state. Okay, so when that complex is not phosphorylated, the PFK2 side is active. Okay, um, some things that make this active, insulin has the effect of dephosphorylating this and making the PFK side active. So insulin will make that PFK side active, which in turn makes F26BP, which in turn turns on glycolysis. Okay. Right, so that increases the F26BP concentration, which stimulates glycolysis, inhibits gluconeogenesis. Okay. The opposite effect, actually phosphorylating this, turning PFK2 off and activating FBPase2, that's done by a hormone called glucagon. Okay, you can kind of think of that as having the opposite effect of insulin. Okay, that creates a cascade that uses ATP to phosphorylate this. Okay, and the enzymes that do that are shown here. Probably not too important that you remember those enzyme names, honestly, for this. But just understand that when it's phosphorylated, the phosphatase is active. When it's not, the kinase is active. Okay. So glucagon and also cyclic AMP concentrations will also go to turn on the phosphatase activity. Okay. When that's active, the phosphatase, you'll get a drop in the fructose 2,6-bisphosphate concentration, which inhibits glycolysis and stimulates gluconeogenesis. Okay, so this is kind of a cool way where this one enzyme complex can control two pathways, right? The glycolysis and gluconeogenesis. So that's, I think, one of the most important when we're talking specifically about regulating glycolysis and gluconeogenesis. That step 3 of glycolysis is very important to remember, and the activity of fructose 2,6-bisphosphate on both pathways is important to know. Okay. The last regulatory step in terms of glycolysis is step 10, and that enzyme, remember, is called pyruvate kinase. It's allosterically activated by fructose 1,6-bisphosphate, so that's the glycolytic intermediate. Okay, so that, again, you can kind of think of that as it's not feedback inhibition, but it's feed forward activation. Right, if you have a lot of that being produced in glycolysis, then you're going to say, oh, hey, we're going to need step 10 to turn on, right? We're going to have a lot of stuff coming through glycolysis. We're going to need to turn that on. Okay. It's allosterically, pyruvate kinase is inhibited by signs of abundant energy, just in general. Okay, and those signs of abundant energy are ATP, having enough of that around. Acetyl-CoA and long-chain fatty acids are other signs of abundant energy. And then also alanine, that's an amino acid. And when you have a lot of that around, that's just is a signal that you have enough amino acids. Okay. So all of those are signals of abundant energy in a cell. Okay. It's inactivated by phosphorylation in response to signs of glucose depletion. So in this is in liver only, the hormone glucagon will lead to phosphorylation of this enzyme. And why that's important? Well, you want to stop glycolysis in the liver and transport glucose from the liver to other important organs that really only use glucose, and that would be the brain. Okay. Right, so this is kind of showing that basically what was in words on that previous slide. In all tissues, we have that fructose 1,6-bisphosphate, okay, the intermediate of glycolysis, not to be confused with F26BP, right? That feeds forward and turns on pyruvate kinase, right? Because if you have a lot of this being produced, you're going to have a lot of, ultimately, you've gone through the commitment step of glycolysis. You're going to have a lot of PEP to get to pyruvate in that last step. So you want to turn that enzyme on. Okay, and then again, you have the signals that you have enough energy in a cell, like obvious one is ATP and acetyl-CoA, that will turn that enzyme off. And and the other things are long chain fatty acids and the amino acid alanine turn that off as well. Okay. And then liver only, you have glucagon, which creates a phosphorylation by protein kinase A that makes this inactive in the liver. Okay. Again, stopping the liver from consuming glucose because that glucose is more important elsewhere, and specifically in the brain, which functions primarily on glycolysis. So the last, the product of glycolysis, pyruvate, can have different fates. Okay, pyruvate can be a source of new glucose through gluconeogenesis, right? So if you have lots of energy, you can take your pyruvate and feed it back into gluconeogenesis to make glucose, which ultimately is stored as glycogen. Okay. You can also use that glucose that you've made from pyruvate to generate NADPH in the pentose phosphate pathway. Okay, so that's pyruvate as a source of glucose. Pyruvate can also be a source of acetyl-CoA. Okay. So acetyl-CoA can be stored as energy in body fat if you have enough energy. It can also be used to make ATP via citric acid cycle if you still are energy deficient. Okay. Acetyl-CoA stimulates glucose synthesis by activating pyruvate carboxylase. So abundant acetyl-CoA means you don't necessarily need energy, and you're going to synthesize gluconeogenesis by activating pyruvate carboxylase. And remember, pyruvate carboxylase is that first step in gluconeogenesis. Okay, so this figure just kind of shows that the alternative fates of pyruvate. Okay, you go through the pyruvate dehydrogenase complex, which makes acetyl-CoA, and we'll talk about this direction of this, the fate of pyruvate, this side, in chapter 16 after the exam. Okay, so pyruvate in the next step in metabolism gets converted to acetyl-CoA. So acetyl-CoA, if that's in abundance, what that does is it turns off the pyruvate dehydrogenase complex and also turns on pyruvate carboxylase, right? And that's the other alternative fate for pyruvate, is going to make glucose in gluconeogenesis. Right. The last few slides here are an examples of gene transcription regulation. And so this table from the text shows the genes that are regulated by insulin. And we talked about insulin a little bit in chapter 12. And one of the reasons insulin is so complex is you can see that there's a number of different genes that are regulated by insulin. Okay, the genes that are increased in expression are generally those involved in glycolysis, right? Hexokinases, phosphofructokinase, pyruvate kinase. So all of those regulated steps are increased by insulin. the expression of those genes. And then you also have that PFK2 FBBase. That's also has increased expression with insulin. Okay, pentose phosphate pathway genes are expressed higher. Fatty acid synthesis. Okay, as you'd expect. If you have the signal of insulin, you want to get glycolysis going. You want to use up some of that blood sugar to make energy or to make other molecules. Okay, what you have decreased expression of is enzymes in gluconeogenesis, and then an enzyme that cuts off, that breaks down glycogen and releases glucose into the blood, right? So if you have insulin, that means the blood sugar has gone up. So you don't want to be wasting your glycogen stores because you have enough glucose in the bloodstream. Okay, so it's kind of a neat example of a signaling molecule we talked about and seeing how it affects some of these metabolic pathways. Not going to be tested on it though, so this is sort of just for your information. Okay. In one specific pathway that the book shows here is by how transcription is activated is this protein cherub. Okay, and it's in response to a higher flux through glycolysis, right? It eventually goes into the nucleus and forms a complex that increases the expression of genes. Okay, not going to be responsible for this on an exam. Here's just another example of how transcription is affected in response to, in this case, insulin, all right, by this FOXO1 transcription factor. Okay. Not going to be tested on this, but this is something that, as the example shows here, insulin signal will definitely eventually cause a cascade where you get an activation of this transcription factor, or it should say inactivation of the transcription factor by protein kinase. Okay, it's phosphorylated and then that's broken down. Okay, without that insulin there, right, FOXO1 would be active and it'd be in the nucleus trying to express these genes for gluconeogenesis. Okay, so you have a lot of insulin, there's glucose around. When you don't have that insulin signal is when you want to be expressing the genes for gluconeogenesis. Again, not going to be tested on any specifics of this. And then finally, I put this in here because this shows you just how complex this can get. Okay, you have these, and these are sites in the genome. These abbreviations that are listed here in this line are binding sites in the genome that come before a particular gene. And I don't remember off the top of my head what gene is actually after this. But these are the transcription factor binding sites. And then all the transcription factors you can see are these kind of figures, blobs that are above. Okay. So think about how complex this is. Some of these, will activate the transcription, others might inactivate it, and the interplay between which ones are around and which ones aren't gets so complex. So I just wanted to include this to show you an example of just how complex some of this regulation in terms of regulating transcription is, because you have so many different transcription factors for some of these genes, and how they interact together is sort of mind-boggling. A little bit simpler example is here in a bacterial protein expression system, where you have a regulatory gene, a promoter, and an operator region. And then you have, if this is an engineered plasmid that you put in a bacteria, then you have some genes that you would, in this case, have genes that utilize the protein products they make, utilize lactose. Okay, and so your RNA polymerase can't attach to the promoter because you have a repressor that is bound to that site before the promoter. Okay, so you have a gene that makes this protein that binds to the operator, and it represses it. Okay, so you can't get transcription of your genes. Okay. In the presence of lactose, though, what that does is lactose will bind to the operator, and when it does that, it removes it. Then that opens up the promoter site for your RNA polymerase to bind, and then you can get the expression of the genes that utilize lactose. Okay. If you instead of putting genes that utilize lactose, if you put a specific gene you want to overexpress in a bacteria cell there, you can turn on that expression, grow the cells up to a very high number, and then turn on the expression of that gene by putting in an lactose analog, something like IPTG, that will bind to that repressor and also does not get metabolized by the cell. And then you can start producing mini copies of a gene that you're interested in. So that's something that has been used in laboratory settings for a very long time to have bacteria make proteins of interest. Okay. So in summary, what we've learned in Chapter 15, hopefully you've learned some general principles about metabolic regulation and how they apply specifically to glycolysis and gluconeogenesis. And really the important steps to remember are first step of glycolysis, third step of glycolysis and its corollary in gluconeogenesis, and then the last step, step 10 to glycolysis.